Welcome to the class of today. For this class, you are going to need make use of your reference textbook, which is a biological science, your biology notebook, and an exercise book in which you'll be doing a series of exercises. We are going to begin with an exercise. State 7 characteristics common to all living things, and for each characteristic, say one way in which it is important to living things. This should be done in the notebook meant for exercises. You must have noticed that one of the characteristics common to all living things is the fact that all living things carry out nutrition. Nutrition is important because it provides energy to living things. Open to page 196 of your biological science and copy the uses of energy into your notebook. This is another exercise that is supposed to be done. This exercise is on types of nutrition. There are several types of nutrition carried out by different types of living things. We have chemosynthesis, photosynthesis, saprophytic nutrition, parasitic nutrition, holozoic nutrition. Draw this kind of table in your exercise book, and for each of the organisms displayed, you put the number, identify the organism, that is, you write the name of the organism on the table, and you place a tick in the appropriate box that corresponds to the kind of nutrition carried out by the organism. So for each organism, put the number, the name of the organism, and place a tick either under chemosynthesis, photosynthesis, saprophytic, parasitic, or holozoic nutrition, depending on the kind of nutrition carried out by the organism. These are the various organisms. Alright, now listen to and copy this into your books, into your notebooks. Subheading: Definition of nutrition. Nutrition is the process by which living things obtain and make use of energy-rich compounds. Nutrition is the process by which living things obtain and make use of energy-rich compounds. There are two main types of nutrition: autotrophic and heterotrophic nutrition. Number 1, autotrophic nutrition. Sometimes called holophytic nutrition. It involves the synthesis of organic compounds from inorganic raw materials. It is used by plants and certain protists and some bacteria. It is used by plants and certain protists and some bacteria. There are two types of autotrophic nutrition. That is photosynthesis and chemosynthesis. A. Chemosynthesis. It is the conversion of carbon compounds and other molecules into organic compounds. In this biochemical reaction, methane or an inorganic compound such as hydrogen sulfide or hydrogen gas is oxidized to act as the energy source. Organisms that obtain energy by oxidation of electron donors are called chemotrophs. If the molecules are organic, the organisms are called chemoorganotrophs. If the molecules are inorganic, the organisms are termed chemolithotrophs. In contrast, organisms that use solar energy are called phototrophs. Subheading: Chemoautotrophs and chemoheterotrophs. Chemoautotrophs obtain their energy from chemical reactions and synthesize organic compounds from carbon dioxide. The energy source for chemosynthesis may be elemental sulfur, hydrogen sulfide, molecular hydrogen, ammonia, manganese, or iron. Examples of chemoautotrophs include bacteria and methanogenic algae living in deep sea vents. The word chemosynthesis was originally coined by Wilhelm Pfeffer in 1897 to describe energy production by oxidation of inorganic molecules by autotrophs. Example of chemosynthesis. In addition to bacterial and archaea, some larger organisms rely on chemosynthesis. A good example is the giant tubeworm, which is found in great numbers surrounding deep hydrothermal vents. Each worm houses chemosynthetic bacteria in an organ called a trophosome. The bacteria oxidize sulfur from the worm's environment to produce the nourishment the animal needs. Using hydrogen sulfide as the energy source, the reaction for chemosynthesis is as shown. This is much like the reaction to produce carbohydrate via photosynthesis, except photosynthesis releases oxygen gas, while chemosynthesis yields solid sulfur. The yellow sulfur granules are visible in the cytoplasm of bacteria that perform the reaction. Definition of photosynthesis. Photosynthesis is the production of food by green plants from carbon dioxide and water using the energy from sunlight, which is trapped by chlorophyll. The carbon dioxide comes from the atmosphere, while water is absorbed from the soil. An equation which sums up this process, though not a very accurate representation of the many steps which actually takes place, is given below. An equation which sums up this process, though not a very accurate representation of the many steps which actually take place, is given below. Equation for photosynthesis. Subheading: Fate of photosynthetic products. Fate of photosynthetic products. The glucose formed by photosynthesis is used as the raw material for other chemical reactions. It is the main substrate used in respiration. It is the main substrate used in respiration. Point 2. Some of the glucose is converted to other carbohydrates, e.g., cellulose, used to form cell walls. Sucrose, to be transported to other parts of the plant. And starch for storage. Three. Some of the glucose is combined with minerals from the soil to make proteins and other complex organic substances. Lastly, the oxygen released is being used by other living organisms for respiration. It is equally used for combustion. Exercise. Find out and write down the meanings of the following words in relation to photosynthesis. The words are ADP, ATP, autotrophs, Calvin cycle, carbon dioxide, carbon fixation, chlorophyll, chloroplast, G3P, glucose, granum, light, reduction, rubisco, thylakoid, lumen. Light harvesting complexes or photosystem complexes. Light reactions, mesophyll cell, NADPH, oxidation, oxygen, palisade mesophyll, Pigalle, photosynthesis, pigment, fate. Okay, copy the following into your notebooks Subheading: Leaves. Leaves can be found in a variety of shapes and sizes. Most leaves are broad, flat, and typically green in color. Some plants, such as conifers, have leaves that are shaped like needles or scales. Leaf shape is adapted to best suit the plant's habitat and maximize photosynthesis. Basic leaf features in angiosperms (in brackets, flowering plants) include the leaf blade, petiole, and stipules. External structure of a leaf. Draw the following structure into your notebook and name the parts. The blade. The blade is the broad portion of a leaf. Apex. Apex is the leaf tip. Margin. The leaf edge boundary area. Margins can be smooth, jagged or toothed, lobed, or parted. Veins. These are vascular tissue bundles that support the leaf and transport nutrients. Midrib. It is a central main vein arising from secondary veins. The base. This is the area of the leaf that connects the blade to the petiole. Petiole. This is a thin stalk that attaches the leaf to a stem. Stipules. These are leaf-like structures at the leaf base. Exercise. Collect fresh leaves from plants with different types of margins, bases, and apices, and fix them with labels on A4 size papers or cardboards, which will be fitted into your biology lab manuals as part of your practical work. Leaves of different shapes and sizes with different types of margins, which could be lobed like this one here. Collect a variety, as many as possible, and display them according to types of apices, types of bases, types of margins. You could decide to show or expose any features of a leaf. Internal structure of a leaf. Subheading. See figure 7.5, page 200 of Biological Science and draw from there. This one is more of a model of the internal structure of a leaf, a magnified model. A model is a true representative of anything. This is a diagram of a palisade cell from a dicotyledonous leaf. A palisade cell from a dicotyledonous leaf. Also draw this into your notebooks. The parts labeled from top to bottom are cell wall, cell surface membrane, cytoplasm, chloroplast, tonoplast. Tonoplast is the membrane of the vacuole. Then we have, after tonoplast, vacuole, and then the nucleus. Diagram of a palisade cell from a dicotyledonous leaf. Draw it in your notebook. Description of the internal structure of a leaf. You're writing. Leaf tissues are composed of layers of plant cells. Different plant cell types form three main tissues found in leaves. These tissues include a mesophyll tissue layer that is sandwiched between two layers of epidermis. Leaf vascular tissue is located within the mesophyll layer. The epidermis. The outer leaf layer is known as the epidermis. The epidermis secretes a waxy coating called the cuticle that helps the plant retain water. The epidermis in plant leaves also contains special cells called guard cells that regulate gas exchange between the plant and the environment. Guard cells control the size of pores called stomata, singular stoma, in the epidermis. Opening and closing the stomata allows plants to release or retain gases, including water vapor, oxygen, and carbon dioxide, as needed. the mesophyll layer. The middle mesophyll leaf layer is composed of a The middle mesophyll leaf layer is composed of a palisade mesophyll region and a spongy mesophyll region. Palisade mesophyll contains columnar cells with spaces between the cells. Most plant chloroplasts are found in palisade mesophyll. Chloroplasts are organelles that contain chlorophyll, a green pigment that absorbs energy from sunlight for photosynthesis. Spongy mesophyll is located below palisade mesophyll and is composed of irregularly shaped cells. Leaf vascular tissue is found in the spongy mesophyll. Vascular tissue. Leaf veins are composed of vascular tissue. Vascular tissue consists of tube-shaped structures called xylem and phloem that provide pathways for water and nutrients to flow throughout the leaves and the plant. Exercise. Identify the parts shown on the following diagrams. Write the name of the figure and then the letters that represent the various parts with the names of the parts. Figure 1.1 shows a transverse section through a dicotyledonous leaf. Identify the parts A, B, and C. Figure 4.1 is a diagram of a section through the undersurface of a dicotyledonous leaf. Identify the parts A and B. Figure 8.1 shows a scanning electron micrograph of a section through a leaf of the Christmas rose, Helleborus niger. Identify the parts M and N. M and N. Copy and complete this table. A palisade mesophyll cell is adapted to carry out photosynthesis. The table below lists some of the adaptations of a palisade mesophyll cell. Complete the table to show how these adaptations help the cell to carry out photosynthesis. First column, adaptation. Thin cell wall, cylindrical shape, large vacuole, chloroplast can be moved within the cell. Column 2, heading, how the adaptation helps photosynthesis. Copy and complete this table appropriately. Adaptations of a leaf for photosynthesis. Adaptations of a leaf for photosynthesis. Point 1. The leaf is broad and flat in shape. This offers a large surface area for the absorption of carbon dioxide and sunlight. Point 2. The leaf is thin. This reduces the distance that carbon dioxide has to diffuse through from the stomata to the mesophyll cells. Next point. The leaf has large air spaces in the spongy mesophyll layer. These provide an easy passage for carbon dioxide. Next point. Palisade cells are at right angles to each other for maximum absorption of sunlight. Next point. The cuticle is transparent for the penetration of solar energy for photosynthesis. Next point. The waxy cuticle is waterproof, so prevents drying up of the mesophyll cells in case of excess heat. Next point. The palisade cells contain more chloroplast, which trap enough sunlight. Next point. An extensive network of veins supplies water to the mesophyll cells and carries the products of photosynthesis from them. Last point. There are numerous stomata on both surfaces of the leaf for the exchange of carbon dioxide and oxygen with the atmosphere during photosynthesis. An experiment to show the distribution of stomata on the upper and lower surfaces of the leaf. Two methods have been described here. Method 1. Peel off the upper epidermis of a leaf and mount on a microscope. Also peel the lower epidermis and mount. To see the distribution on the two surfaces. Method 2. Take two leaves of the same plant and of the same surface areas. For one, seal the upper surface with aluminum foil, and for the other, seal the lower surface and allow transpiration to take place. The one with the upper surface sealed loses more weight than the one with the lower surface sealed. Assignment. The two methods described in the experiment above have not been described following the scientific method. Re-describe the above experiment properly following the scientific method. That means use the following subheadings to describe the two methods. Begin with the aim, requirements, procedure, observation, and conclusion for each. This should be done in your biology notebook as part of your notes. Subheading: Structure of chloroplast. See and draw figure 7.7, page 201 of Biological Science. This is a model of a chloroplast. You can look at it closely. This is another representation of a section of a chloroplast. Description of the chloroplast. Copy. Chloroplast structures include 1. Membrane envelope. Contains inner and outer lipid bilayer membranes that act as protective coverings and keep chloroplast structures enclosed. The inner membrane separates the stroma from the intermembrane space and regulates the passage of molecules into and out of the chloroplast. Intermembrane space. This is a space between the outer membrane and the inner membrane. Thylakoid system. Internal membrane system consisting of flattened sac-like membrane structures called thylakoids that serve as the sites of conversion of light energy to chemical energy. Thylakoid lumen is a compartment within each thylakoid. Grana, singular granum. Densely layered stacks of thylakoid sacs, 10 to 20, that serve as the site of conversion of light energy to chemical energy. Stroma, dense fluid within the chloroplast that lies inside the envelope but outside the thylakoid membrane. This is the site of conversion of carbon dioxide to carbohydrates or sugar. Chlorophyll, a green photosynthetic pigment within the chloroplast grana that absorbs light energy. Exercise. Identify the parts indicated on the following diagrams. Figure 1.1 shows an electron micrograph of a chloroplast. An electron micrograph of a chloroplast from a maize leaf cell. Identify the parts X, Y, and Z. State two features visible in the above diagram that identify the organelle shown as a chloroplast. Chloroplasts absorb phosphate ions from the surrounding cytoplasm. Suggests one way in which chloroplasts use phosphate ions. D. Starch grains in plant cells contain both amylose and amylopectin. Explain how both of these substances are formed from glucose in plants. Answer this question in your exercise book. We will end here for today. Thanks for watching. And see you in class.